Hello, and welcome to the webinar, What Everyone with MBC Should Know About Antibody Drug Conjugates. I'm Kate Vieira-Fitzer, the Metastatic Breast Cancer Program Director at SHARE.
And I'm Victoria Goldberg. I'm a host and a producer of the Our MBC Live podcast.
Before the presentation begins, I'd like to tell you a little bit about SHARE. We're a national nonprofit organization that supports, educates, and empowers anyone who has been diagnosed with breast or gynecological cancers. We provide outreach to the general public about signs and symptoms, because no one should have to face breast, ovarian, uterine, cervical, or metastatic breast cancer alone. For more information about upcoming webinars, support groups, and our helplines, please visit our website at sharecancersupport.org.
Before we begin our podcast, we have a few housekeeping reminders for you. All participants will be muted during the presentation. Once the speaker finishes presenting, we'll begin a Q&A discussion. Feel free to ask any question through a Q&A section at the bottom of the screen. Remember that Dr. Hamilton is unable to give specific medical advice, so please keep your questions general in nature. We also have closed captioning available. You can enable this feature by clicking the live transcript button on the bottom of the screen and selecting the subtitle option.
This webinar is heavy in science, but hang in there. The webinar is being recorded. We will turn this into a podcast and we'll break down the difficult parts for a better understanding. Once again, listen to get an overall picture, but don't worry about the details. We'll share the recording in a few weeks with all registrants, and it will also be added to our website. Dr. Hamilton.
Hello, I am Erika Hamilton. I am at Sarah Cannon Research Institute. I lead our breast cancer program here. I've been here for a little over a decade. I'm very eager to talk to you guys. So if you guys want me to start screen sharing my slides, can you give me permission for that? Let's see. How's that? Can you guys see?
Perfect.
Okay, perfect. So again, I'm going to be talking about antibody drug conjugates. And as warned, this is a little heavy in science. I'm going to try to make it digestible into some simple bites. But I'm going to also give you enough information that if you want to come back later and dive in a little bit more, it'll be there. This is a super long list of conflicts of interest, and you'll note that they all say to institution only. I take no personal funds from pharma. So we can all debate whether that's really a conflict of interest, I guess. Okay, so antibody drug conjugate. What are these? What on earth is an antibody drug conjugate? And so the big thing to remember about these is it's three parts. So the Y, big Y in the middle of your screen, that's the antibody. This is the targeting, right? So you can think of trastuzumab, which is Herceptin as an antibody, pertuzumab, as an antibody. Then you have this linker, which are these little orange bars sticking out the side where the stars are. That's what links the chemo, that's the star, a cytotoxic payload. So how I like to explain this to my patients is it's not really chemo. It's not naked chemo in the way that you think we infuse chemo in the body. It goes everywhere, kills cancer cells, but it also kills a lot of healthy cells, which leads to nausea, hair loss, neuropathy, all the side effects we get from chemo. This is kind of smart bomb chemo. So it's only going to the cells that express that antibody. So for example, a HER2 antibody drug conjugate is targeting HER2 cells. When it binds, it dumps the chemo right on that cell. And for a large part, it's kind of sparing the rest of the body. So we definitely have side effects with antibody drug conjugates, but they are not necessarily the traditional side effects that you think of with chemotherapy. So this is essentially how it works. This antibody goes in, it binds to its target on the cell. When it binds, it gets taken up by the cell into this little internalization vacuole. The pH in these vacuoles change, and that's actually what allows the linker to dissolve, and it releases its chemo. The chemo is then inside the cell, causes the cell to die. And what's also special about these is some of them have something we call bystander effect. And what that means is it not only affects the cell it explodes in, but a little bit of that chemo can leak out to maybe all the cells that touch that cell. And that becomes really important when we're talking about heterogeneity or low expression of the antigen. And the classic example of this is trastuzumab deruxtecan or Enhertu, where even when HER2 expression is really low, this drug still works very well. So what's our history with antibody drug conjugates? A lot of people think, oh, well, these are brand new and they're all kind of taking breast oncology by storm. And for a large part, they kind of are taking breast oncology by storm. But remember that we actually had an FDA-approved antibody drug conjugate for breast cancer over a decade ago, and that was TDM1 or Kadcyla. Now, this drug behaves very differently than the ones that we're giving now, but that was chemo that was linked to trastuzumab. So it was an antibody drug conjugate. Now, when we get to 2019, in the past couple of years, we had approval of trastuzumab deruxtecan, which is HER2 targeting. We've had approval of sacituzumab, which is TROP2 targeting. And then you're going to hear a lot about other ones, Dato-DXd, which is another TROP2 targeting one. We also have a HER3 DXd, which is called patritumab deruxtecan. There are a lot of these drugs currently in clinical trials. So first, let's talk about the original TDM1. Okay, so this is a HER2 antibody that's essentially attached to a maytansinoid payload, and you can kind of put this kind of in a taxane bucket. So you can kind of think of it as like a paclitaxel. So this was based on the Amelia study initially, and as well as Teresa. So Amelia compared essentially third-line HER2 positive therapy, which was capecitabine lapatin at the time, to TDM1, and you can see, you know, about a 50% improvement in progression-free survival, 6 months up to 9.5 months. Teresa was TDM1 versus treatment of physician's choice. And again, you see about a doubling in progression-free survival, 3 up to 6 months. So TDM1 was approved initially for patients that had HER2 positive breast cancers that had already seen trastuzumab and a taxane. Now, ultimately, TDM1 also got approved in the adjuvant setting. So what's the difference between TDM1, which was also an antibody drug conjugate, and these newer antibody drug conjugates where we talk about bystander killing effect? What you can see here is that when this is uploaded into this number 3, into this vacuole, when it pops out and the little orange ball chemos kill the cell, when that cell dies, it pops and it lets a little bit of the chemo out. That's not going to go from, you know, your lung, your right lung, all the way to your brain, right? This is just a controlled bystander effect. So it's going to touch the cells that were right around that cell that it died. And so that's really what's enabled us to use antibody drug conjugates where the expression isn't high. Because TDM1 is certainly not a drug that we use for HER2 low or HER2 zero. You have to really have cancer that's HER2 driven for that to work. And these newer antibody drug conjugates are a little bit different. So the first one was trastuzumab deruxtecan. You guys might know this is in HER2, but I try not to use trade names. So again, it's a HER2 antibody. It's really trastuzumab, right? And now our payload is a topoisomerase inhibitor. So exatecan, you can think of this as like when we hear chemos like irinotecan for GI cancer, et cetera. And then it has a cleavable linker. So what's unique about this is it has a much higher drug to antibody ratio. So how many burgundy little balls are hanging off of one HER2 antibody? TDM1 was quite low. Here, there's 8 chemo molecules on one antibody. So it has a higher DAR, you may hear people say, drug to antibody ratio. It also has a bystander effect. So here's a little chart. You can come back and look at this. So bystander killing effect, yes with TDXD, no with TDM1. Targets HER2 positive tumors, yes to both. Targets HER2 low, only TDXD. So what about sacituzumab govitecan? You might call this SASI, you might call this Trodelvy. You probably don't say sacituzumab govitecan, but I've learned how to do it well. And so this is a TROP2 directed antibody drug conjugate. So TROP2 is the part of the molecule that's like HER2. It's finding a TROP2 expression on a cell, it's binding it, and then it's releasing its chemo there at that cell. This has an SN38 payload, so it's kind of in the same family, irinotecan-like. And again, it has a cleavable linker. You can see that the DAR here, 7.6 to 1 on average, is pretty similar to trastuzumab deruxtecan. Both of these have higher DARs than TDM1. This is now approved for both triple-negative breast cancer as well as hormone receptor positive breast cancer. And so this has actually been a big paradigm shift for us in the clinic and for you guys as patients, because we're very used to a drug having a certain kind of breast cancer that it's approved for. And a lot of these antibody drug conjugates can kind of go across HER2 high and HER2 low, or triple negative and hormone receptor positive. So the swim lanes of kind of having 3 different types of breast cancer isn't quite as clean as it used to be. So let's go through a little bit of an algorithm if we think about HER2 positive. Right now, the standard for first line is still taxane, which is naked chemo, with trastuzumab and pertuzumab, 2 antibodies against HER2. There are multiple trials challenging this first line. We're looking at trastuzumab deruxtecan, we're looking at new combinations, et cetera. But right now, that's the standard, is that people get chemo with trastuzumab and pertuzumab. In the second line, based on DB03, TDXD is probably the standard. Capecitabine, trastuzumab, hidacatinib are probably third. It does have an approval in second line for patients that have brain metastases. And then fourth line and later is a little bit of the wild, wild west. We have a lot of agents. And to be quite honest, because tucatinib's newer and because trastuzumab deruxtecan is newer, we honestly don't know which of these regimens really performs the best after these 2 treatments, because they were approved before the second and third line drugs even existed. So we've got TDM1 we can come back to. We have neratinib and lapatinib, our TKIs. We have that new antibody margetuximab that we can give with chemo. And certainly we have a ton of agents in clinical trials. So I'm going to give you a little bit of definition refresher before we dive into these studies, because you're going to think that I'm just throwing words at you that mean nothing. So ORR is overall response rate. That's the percentage of people that have their tumor shrink by at least 30% when they get the drug. Okay, so if their tumor shrinks by 10%, that's kind of still considered stable disease. So response rate is, in my opinion, pretty impressive. It's a 30% shrinkage of the lesions they had. Duration of response is of that response and them doing well on the drug, how long did it last? Progression-free survival is how long they're on a drug before the drug stops working and their cancer grows. Okay, so you can imagine duration of response and progression-free survival are a little bit similar, but duration of response refers just to those people that were responders, whereas PFS includes everyone. Okay, and then overall survival, how long did they live? That's the gold standard in breast oncology, although it's a little bit trickier to hit than some other tumor types like pancreatic, where people only live a year. So if you've got a good drug, it's pretty easy to show a difference there. If you're looking at a drug for hormone receptor positive disease, where people might live 5, 8, 10, 12 years, one drug to change how long somebody lives is a little bit of a higher bar. Okay, so this was the initial trial with trastuzumab deruxtecan. It was a phase 2 trial. And on the left, you see what we call of a waterfall plot. And so grossly, you can look at this curve, and if about half the people are going down, the bars are going down, and about half is up, it means it works for about half the people. When you look at this, you'll notice there's 1, 2, 3, 4 people out of 168 that had their cancer grow. Everybody else had their cancer shrink, and a lot of people had their cancer shrink a lot. So the overall response rate was over 60%. It lasted a really long time, over a year. And remember, these were pretty heavily pretreated patients. And what's the most common side effects? It's ILD, it's nausea, and a little bit of heme toxicity. So based on this, it's hard for me to believe that this has been approved since 2019. It was right before Christmas. I remember it. And this was approved for patients with HER2 positive metastatic disease after 2 prior anti-HER2 regimens. It's now approved after 1, but we'll get there. So this is Destiny Breast 03. This is more recent data. So this was the phase 3 trial trying to move TDXD from third line into second line. And what was our second line regimen? It was TDM1 at the time. So head-to-head trial, people either got trastuzumab deruxtecan or they got the standard at the time that was TDM1. Everybody had already had pertuzumab. So that classic trastuzumab, taxane, and pertuzumab. And this was what we saw. The dark blue line is trastuzumab deruxtecan. The gray line is TDM1. And the reason the dark blue line is on top is more people didn't have their cancer grow for longer. And so you can see there's a big gap between these lines. We like to call these progression-free survival Kaplan-Meier curves. If you can drive a truck between the two lines, you're doing well. And I definitely think we can drive a vehicle through this line, okay? Now, if you look over on the right, this is overall response. And so again, if you say, okay, where's the break point between people that had their cancer grow and people that had their cancer shrink, you can see that the dark blue lines, many more people had their cancer shrink there compared to TDM1. It was almost a guarantee that at least a scan was going to look pretty good. And so this became now our second line regimen approved for people that have had at least one prior HER2 regimen. Okay, so where's the twist come in? The twist comes in the fact that always we had given people that had high HER2 expression, HER2 targeting drugs, and we didn't give it to people that didn't have HER2 high. Not because we didn't look, but because it didn't work. We did a big study with trastuzumab, Herceptin, because it was so helpful for patients that had high HER2 expression. And it was completely stone cold negative in people that were HER2 low. It wasn't like we didn't look. It didn't work. It was a different type of drug. But now things have changed a little bit. So when we say HER2 low, what are we talking about? I'm not talking about people that are 0 on the left where there's no expression. I'm talking about these people in the middle with 1+ or 2+ expression. So it's not HER2 positive. It's not HER2 0, but it's HER2 low. There's a little bit there, but it's not a lot. So how common is that? Super common. It's more common than being HER2 positive. And if you look at hormone receptor positive disease, the green part of the pie is an IHC of 2+. The red part of the pie is 1+. So together, these are HER2 low. So you can see that 75% of people are HER2 low, only a quarter are HER2 0. Now, when you go over to triple negative, it's a little bit less, but still 50% of people essentially are 1+ or 2+ in low, and half are 0. So you can see the majority of hormone receptor positive disease actually has HER2 low expression. And so what's our activity here? On the left, you can see all patients. Red bars are 1+, blue bars are 2+. And you kind of see how the red and the blue plot out. And I don't see a big trend there. And if we look over, was it just the 2+ patients? Was it just the patients that had higher HER2 expression that did well? No, there were plenty of people in red, 1+ over in C, all the way on the right that did well. So it doesn't take a whole lot of HER2 expression to have activity of this drug. And even when we talk about people that are HER2 negative and have HER2 low expression, the overall response rate is almost 40%. And the disease control rate, meaning you got to a scan and your cancer was not growing, was approaching 90%. So this is Destiny Breast 04. This was the registrational trial. And they said, this data looks pretty good. Let's put it in a bunch of people that have some HER2 low expression. So 1+ or 2+. And we're going to compare it to whatever they would normally get, which certainly isn't a HER2 targeting drug. It's chemo. So capecitabine or eribulin or gemcitabine or paclitaxel or nab-paclitaxel. And some of you may have received some of these agents. Now, how did the population split out? The majority of people were hormone receptor positive in their cancer, which shouldn't be surprising because HER2 low is more common in hormone receptor positive. And about 2/3 of people had already seen at least 3 lines for metastatic disease. And about 2/3 had already seen CDK4/6. Okay, 70% of those patients that were hormone receptor positive. And the reason it's not 100 is this was enrolling around the time that CDKs came out. So it's a little bit of a miss, and not all countries in the world have CDK access. So what did we see? On the left, for patients that were hormone receptor positive, you see these curves looking pretty separate. When we look over on the right, and now it's all patients, so adding back in those patients that had triple negative disease, again, looks pretty good. And in fact, people with triple negative breast cancer tend to do less well in terms of PFS. And so our benefit is almost 5 months in hormone receptor positive disease and almost 5 months in all patients too. When we look at overall survival, our overall survival across the board is half a year longer. So just by giving a drug at this line of therapy, even if they went on to get other therapies after, we can improve overall survival by a half a year. So progression-free survival was about 5 months improved. Overall survival was 6.5, almost 7 months. And so this really became the new standard of care for HER2 low. Now, what isn't this? This isn't, oh my gosh, it's the magic drug. If I have hormone receptor positive disease, I want to get this first. Forget about endocrine therapy. That's not what this is. All of these patients that had hormone receptor positive disease had already received lines of endocrine therapy until they were endocrine resistant. You want to be systematic and march through your endocrine therapies first, and then they'd had a line of chemotherapy. Then they went on to this. So most people had capecitabine first line. It tends to be the drug we give first. Now, what about the sacituzumab govitecan? You're going to see the trial design looks very similar. Now we're talking about the TROP2 antibody drug conjugate. These patients are HER2 negative and they have hormone receptor positive breast cancer. And again, very similar. They either get sacituzumab. Now this drug isn't every 3 weeks. It's day 1, day 8, and day 15 off. So you get it for 2 weeks, you get a week off, and it's every 3-week cycle. And we compared against chemo again. Significantly improved progression-free survival and overall survival, although the actual number benefit wasn't as quite as high as we saw with trastuzumab deruxtecan. So where do we currently sit for hormone receptor positive HER2 negative disease? So you see first line, still endocrine therapy with the CDK4/6, okay? A lot of trials have an overall survival benefit there. Second line kind of depends on what flavor of cancer you have. If you have a BRCA alteration, maybe a PARP. If you have an ESR1 mutation, we now have elacestrant approved. If you have a PI3 mutation in your cancer, that's somewhere where we think about alpelisib. If you don't have any mutations, but you did well on endocrine therapy CDK, I'm probably going to go in with another endocrine agent. And outside of a trial or a mutation, that could be fulvestrant with everolimus. If people don't do well to CDK, that's where we'll be thinking about chemotherapy. So you're going to continue to get endocrine therapy until progression again. Then you think about chemo, and then this is when the antibody drug conjugates would come into play. Now they're both kind of approved in the same line. So it's up to individual patients and individual physicians and the different side effects between them, what they may want to receive. And we're going to go into that a little bit. So this was ASCENT. This is the phase 3 confirmatory trial. So now we're talking about triple negative breast cancer. Okay. And again, it's confusing. It's the same drugs. We're talking about sacituzumab again, and it's practically the same treatment design. So people had to have had at least 2 chemos in the metastatic setting, one of which could have been early if they relapsed quickly. They either got chemo or they got sacituzumab, and this was the benefit. This was statistically significant. So we can see progression-free survival from 1.7 months up to 5.6, and overall survival from half a year up to a year. Now, what stands out to me here actually isn't the fact that sacituzumab is better, which it is. What stands out to me is that chemo that we traditionally think of that works in breast cancer is really giving people 2 months without their cancer growing in triple negative breast cancer. Just not good enough, right? So based on this, in 2021, sacituzumab got approval for people with triple negative disease that had seen 2 prior systemic therapy. So one of the big questions is, well, is it like in HER2, trastuzumab deruxtecan? Do you have to have a certain expression of TROP2? And we looked at that. So the gray bars are treatment of physician's choice. The green shaded bars are people that had TROP2. And you can see across the board that regardless of whether people had high, medium, or low TROP2, sacituzumab always beat the standard chemo. Now, it didn't really affect overall response rate that much. It did kind of tend to be that if you had really high TROP2 expression, you probably were going to do better on saci for longer. But even for people that had low TROP2 expression, they were better off getting sacituzumab than they were chemo. And so for that reason, we don't check TROP2. It's eligible for everyone with hormone receptor positive or triple negative disease. So following the data there. So what's our algorithm for triple negative? And this gets a little more complicated. So first line it depends. Is your cancer PD-L1 positive or negative? If it's positive, you're getting chemo with Pembro probably. If it's negative, you're probably getting chemo. Then after that, if you have a BRCA alteration, PARP would be a good idea. And then probably sacituzumab or trastuzumab deruxtecan. And technically these could be given in either order. But remember that in the trastuzumab deruxtecan study, majority of people were hormone receptor positive. So a lot of people think that the sacituzumab data is a little bit stronger in triple negative. But really, I think both of these drugs are very valid options. Okay, so now let's get into, okay, that's great. These drugs really work, but what's the price? What are people having in terms of side effects and what's quality of life like? And I think one of the biggest take home, if you can take away something, is just because you've had one antibody drug conjugate, don't assume the other ones are the same. Or just because you've had one and maybe didn't do well on it, it doesn't mean that you're going to have the same side effects with others. Although these are all called antibody drug conjugates, they are very different drugs. They target different things. They have different chemo payloads. They have different side effects. So TDM1 is hair loss and low platelets. Doesn't have a lot of nausea. Doesn't have a lot of ILD. Doesn't really have a lot of diarrhea. Trastuzumab deruxtecan in the middle, the most common side effect is nausea. And in the trials, we didn't do a great job managing nausea because it wasn't recognized that people had so much nausea. And so no mandatory antiemetics or nausea medicines before we gave the drug was required in the trials. Now it's classified as a moderately emetogenic drug, meaning it causes quite a bit of nausea for people. And so NCCN recommends a 2 or 3 drug nausea regimen. So in my practice, that's something like Zofran on Dancitron, a steroid, and then probably also an NK1, which is a drug like Amend or something like that. A lot of other people kind of use 2 drugs to start and then add Amend if they need it. But I kind of like to go heavy and then peel it back if we're doing well. Because the last thing you want to do is kind of blow somebody out of the water and they're like, I'm not ever taking that drug again. And you're like, oh, if I just added another one up front, right? Sacituzumab, Trodelvy on the right, it has some nausea too, but the biggest side effects are diarrhea and low counts. The diarrhea can be really not very bothersome for some people. I have some people that don't take a single medicine for diarrhea. They don't have diarrhea. And I have other people that are on like 3 medicines for diarrhea. So this really depends on who you are. So if you talk to somebody, they're like, the diarrhea was horrible. Don't assume you're going to get it because it's very, you either have it bad or you don't. It's not that everybody has diarrhea. And then neutropenia. And this can actually be kind of problematic a bit. And that's because the drug's given on day 1 and day 8. And so how disappointed are you when you come in when day 8 and your counts are too low and you can't get your chemo? And so a lot of times we end up having to play with growth factor, but it's tricky because we can't give Neulasta after day 1 because they have to get chemo within a week and it's not approved there. So a lot of times we try to give the day 1, try to give the day 8, give Neulasta after the day 8 because they're not going to get chemo for 2 more weeks and try to ride into the cycle with a really high white count. So it lasts for 2 chemos and we try to go that way. We can also use dose reductions. So the thing that's gotten a lot of attention is ILD or inflammatory lung disease, pneumonitis it may be called. It's patchy inflammation in the lung. And so particularly with trastuzumab deruxtecan and HER2, we have to really watch for this because what happened is before we recognized that this was a big side effect, people actually died from ILD. It's pretty rare to die for ILD now. Knock on wood, I've never had a patient that's died from ILD, and I've been using this drug for a long time. But we have to be pretty strict about it. And so there's not a good screening test. You can't measure somebody's oxygen and be like, I know they don't have ILD. You can't do pulmonary function tests or chest x-ray and say they don't have ILD. It really has to be a CT scan. And so because of this, I keep patients that are on HER2, I keep their scans pretty frequent. In general, about every 9 weeks, because insurance doesn't tend to pay for scans less than 2 months. And because this is an every 3-week cycle, we really can't get them at week 6, but week 9, we can get them. Now, it's tricky because a lot of people don't want to be in the scanner every 9 weeks. And especially if you're going to be on a drug for 2-3 years, which some of my patients that have been on HER2 have been, they don't want to do this. But at the same time, we really need to because I want to catch ILD when it's grade 1 and it's just showing up on a scan. And I don't have to stop their drug permanently. I can give them some steroids, get rid of it, and get them back on drug. because if you show up symptomatic with lung spots or coughing, etc., then you're not supposed to re-challenge with drug. And it's really disappointing when somebody's responding really well and they can't go back on drug. So here's the, and I know this is a very busy slide, but here's the recommendation in terms of nausea medicines. So you'll see dexamethasone, clonazepam, that's that NK1. And then it says you can also add a 5-HT3. Okay, so we're definitely going to leave time for questions at the end, but I want to tell you a little bit about what's coming. Because yes, we have 3 approved antibody drug conjugates. We have the oldie TDM1. We're trying to figure out exactly where we're going to give that. We still give it for adjuvant HER2 positive disease with residual disease a lot, but in the metastatic setting, we're probably giving it more 4th line now. Used to be our 2nd line drug. We have sacituzumab, our TROP2 ADC for both hormone receptor positive and triple negative disease. And we have in HER2, trastuzumab deruxtecan for both people that have HER2 high breast cancer, as well as people that have HER2 low. And that includes both triple negative and hormone receptor positive. So what's coming? The big one, Destiny Breast 09. Answering the question of, is should in HER2 be 1st line? Should we move the taxane, trastuzumab, and pertuzumab? And you'll see this is a little bit of a clever design. Arm C is what we normally give now, taxane, trastuzumab, pertuzumab. Arm A in HER2 alone. And Arm B is a little bit of a combo in HER2, but add the pertuzumab that they otherwise wouldn't be getting. Because remember, part of TDXD or in HER2 is kind of already trastuzumab. So that'll be 3 arms. This is already enrolled. Everyone's on it, and we're just awaiting the results. So we should see this in the next year. Destiny Breast 06. And so this is trying to answer the question of, okay, we give TROP2 to everyone. We don't even test TROP2. In HER2, now you've proven it works for high. You've proven it works for low. Why don't you give it to everyone? Why do we care if somebody is 0? Are they going to respond anyway? And there's some data from a trial called DAISY that suggests, yeah, it might not be as high of a response rate as if you have higher expression, but the response rate looks 30 to 40%. Which is not bad in a disease like triple negative. So this is asking the question of trastuzumab deruxtecan versus chemo for the 1st line of essentially chemo, okay? And so it's enrolling low as well as ultra low, so kind of even some of these 0s. Again, this trial's done. Everybody's already been on it. We're just awaiting results. This is Tropian Breast 02. This is looking at one of those newer antibody drug conjugates. It's not approved yet, Dato-DXd. And so this is a TROP2 antibody drug conjugate, but the payload is like in HER2. So you can kind of think of it as almost a fusion between the SASI molecule and the TDXD molecule. It's got the TROP2 part of SASI, and it's got the chemo part of HER2, okay? And this is for patients that have triple negative breast cancer up front, not even for people that have already failed chemo in the metastatic setting. This is what you get first. And many of you may not have seen this today. There was a big press release from AstraZeneca, not this trial. This was Tropian Breast 02 or TB02, but TB01 in a press release this morning was positive. And that was for people that had hormone receptor positive disease in the 1st line chemo setting. So again, they'd done their endocrine therapies, but when it came time to get chemo, they either got Dato or they got normal chemo, naked chemo. And this was positive for progression-free survival, a trend for overall survival. So we will see this presented at a meeting later this year. I don't know if it'll get into ESMO, but it will by San Antonio. So we're going to see it at one of those. So what about our other TROP2 antibody drug conjugate? This is sacituzumab. So ASCENT-03 on the left is that you're PD-L1 negative or positive and had already gotten Pembro in the neoadjuvant setting. And so it's sacituzumab versus chemo. Or ASCENT-04, which is the other flavor, if you were PD-L1 positive in your tumor and you hadn't seen Pembro, well, we want you to give you immunotherapy. And so it's SASI with Pembro or chemo with Pembro. So 2 flavors because there's kind of 2 different ways we would treat patients based on whether their tumors had PD-L1 positivity. Another antibody drug conjugate that's actually reported quite a bit of data. So this is patritumab deruxtecan. And every time you hear deruxtecan, think it's a cousin to Enhertu, okay? So we've talked about TDXD, HER2 with deruxtecan payload. We've talked about Dato DXD, TROP2 with deruxtecan payload. Now this is a HER3 antibody drug conjugate with deruxtecan payload. So it's the third antibody drug conjugate made by the same company with the same payload. And it has activity across all three types of breast cancer: hormone receptor positive, triple negative, and HER2 positive. And so this is actually a study I'm leading. It's called BRE 354. On the left, you can see we had hormone receptor positive disease and triple negative disease. On the right, in part Z, we have HER2 positive breast cancer. And we're enrolling in expansions right now. And we're specifically enrolling patients that have already seen antibody drug conjugates, because sequencing is going to be a big deal here. Are we going to be able to give the same type of ADC with a different target right after each other? Are we going to have to change the chemo payload? Is there going to be resistance to another one if you've already given one? And so we're specifically enrolling patients that have already seen an antibody drug conjugate to answer this question. I suspect that maybe PFS isn't going to be as long, but I think it's still going to be chemo. So important question. And that's it. So I'm going to stop sharing and I'm going to turn it back over and we'll do some questions. Oh, there's a good question in chat. Can I dive in there?
Oh, good. We don't need to ask any questions. She's going to go and do it herself.
So everyone, you can go ahead. Go ahead.
There is a question in chat that from Blanche that says, is ILD reversible off drug? It's kind of a nuanced question. In general, yes. So I've had multiple people that have had grade 1, grade 2. I've had just a couple grade 3 ILDs. Yes, it's reversible. The problem is that for the more severe cases of ILD, it can take a long time to completely go away. Now, luckily, grade 1 and grade 2, people really don't have a whole lot of symptoms. I mean, for grade 1, people feel fine. They don't even have a cough. They're not short of breath. And I'm telling them, hey, there's something on your CT scan. And they're going, I don't care. This drug's working great. Don't stop giving it to me, right? But I'm worried because once somebody has grade 4 ILD or sometimes grade 3 ILD, it doesn't seem like steroids, et cetera, help as much as we want it to. And so that's where some of these people have actually gone on to get grade 5 or death. And so we know with ILD, if we kind of re-challenge and it's too bad, or we miss it and don't catch it early enough, people can actually die. And so we're really cautious about it. But yes, in general, it's reversible. Sometimes it can be a little bit of a problem for clinical trials because if you think about getting a scratch on your leg, right, and you have a scar, that's essentially what we're looking at as a CT scan. We're looking at a little scar in the lung. And as you guys know, when you have a scratch on your leg, I mean, that scar can be there for a while before it goes away. It gradually fades, it gets better, right? But it can be there for a while. And so a lot of trials, if you have ILD, won't let you go back on until it's resolved. And so sometimes that takes months.
Kate, can I ask a follow-up question?
Mm-hmm.
I have a
you can ask anything you want.
Yeah, no, but I'm following the orders from my management, and Kate is, so I'm deferring to her. So I wanted to ask you this question. It's related to ILD, and actually this was a real life question that was asked at our group. So what about, ILD sometimes is a side effect of Ibrance. Mm-hmm. So some people get ILD when they are on Ibrance, surprisingly.
Yep.
So the question was, if you had ILD on Ibrance, would you be even considered as someone who can, okay.
I would not assume because you had ILD to an endocrine therapy that I wouldn't try Enhertu for a number of reasons. One, sometimes we're kind of bad at saying something's actually ILD. I mean, people have big lungs on both sides and we're looking at some tiny little patchy infiltrate in one spot. Well, we're in the middle of a COVID pandemic. There's a lot of reasons to have a patchy infiltrate in your lungs, right? Not to mention flu or respiratory virus or anything else. So I think if we penalize people for ever having a little ILD, we're going to box people out of a lot of therapy, right? And they're so different. It's different mechanisms of actions for the drug. So because one did it, there's no reason to think that another one does it. You know, it's actually a side effect of a lot of drugs. We didn't even know it was a side effect of CDK4/6 inhibitors. Didn't come out in any of the trials with thousands of people. It wasn't until the FDA pulled across Palbo, across Ribo, and across Abema that they actually saw a trend about, maybe about 2% of people have ILD. It's easier to pick out within HER2 because it's probably about 15%. Now, more severe ILD is less than 2%. But about 15%, 1 in 8-ish, 7-ish people probably will have a little bit of ILD.
Thank you. Okay, actually, you know what? I answered the same thing, and I'm so glad I said the same thing to the person who asked me that question. Go ahead, Kate.
So we're going to go to a submitted question that came in upon registration. So people are wondering, you know, in the clinic, when you're presented, when the patient comes in and you've seen them quite a while and they've progressed on their treatment, how do you determine the best ADC to use? And then, you know, is it important because now we see all these clinical trials coming? You're talking about the HER3 trial and the Dato-DXd trial. Like, when do you consider that clinical trial? Do you consider it at the get-go, or like, how do you think about the individual patient?
So yeah, so my rule of thumb is always consider a clinical trial. It doesn't matter what line you are. It doesn't matter what stage you are. There are neoadjuvant for people that are going to have cured breast cancer. There's adjuvant. There's trials where they just collect blood and it doesn't even have anything to do with you getting a drug. And then first, second, third, at any point, there are clinical trials. Now, what's interesting, you know, is there used to be kind of a perception about, well, you know, clinical trials are just for people that don't have anything left. Why would I want to do a clinical trial instead of a drug that's been proven to work? Well, the data shows when we look across clinical trials that by the time a drug gets to a clinical trial, we are pretty darn sure that drug works. And the trial that it's in is to prove that it's better than something else. So people that participate in clinical trials from phase 1, phase 2, phase 3 actually have longer survival than those who don't. And it's because drug cancer care moves so quickly that essentially, if you're using clinical trial options, you're getting treated with the drugs that are going to be standard in 5 years from now. So I encourage people to look at clinical trials early. And there's another reason that's a little bit self-serving for those people, right? Is that a lot of clinical trials are picky. We all talk about this, right? The FDA is asking for cancer Olympians. They want people that are in great shape. They want perfect kidney function, perfect liver function. They want your counts to be good. And so if you exhaust all your normal therapies and your body's a little bit beat up because we've been giving you poison, right, to treat your cancer, then you might get to go to clinical trial and we're like, you're not eligible. Those numbers aren't good enough. Or the trial only wants people that have had up to 2 lines of therapy and you've had 3. If you use your trial opportunities up front, we as oncologists can always pull chemo off the shelf. We can pull chemo off the shelf even if the liver doesn't look perfect. We can cheat with things. We can dose reduce things. You can't cheat on clinical trials. So in general, you want to use your clinical trials up front. And then you want to start pulling things off the shelf, maybe when you don't qualify for a clinical trial, and that's going to give you more treatment options for longer than if you didn't use them.
So a follow-up to that, you know, whose responsibility is it to find that clinical trial, and how, you know, do you rely on your physician? Do you have to look yourself? Like, it just seems like a complicated process and definitely overwhelming.
So there's not a great answer to that. I would love to say that it's the physician's job. I think it's my job, but the reality is that most oncologists in America don't have access to trials. And not only that, nobody's really educating them about these trials or where to go. There are websites like clinicaltrials.gov, other people like Mediviver or Tiger Lily, have some clinical trials listed on their site. But if you're just not a healthcare trained person, what the information that you can find off of those is just really unsatisfying. You may find a trial, and even if you know all the characteristics of your disease and your organ function and stuff, then you might call them and they say, oh, I mean, that's still online, but we finished enrolling that 3 months ago. So it's just not very good at finding what's really available right then. So this is what I suggest. I don't think it's ever inappropriate to ask your doctor of, hey, is the clinical trial right for me at this one point? And if they look kind of like, I don't know, then just say, hey, could I get a second opinion before I start this therapy just to see if there's a clinical trial for me? If not, and they say that this therapy is what I'm going to do, I'm going to come back and get it with you. But it's always okay to get a second opinion.
Thank you.
Okay, thank you.
Okay, Dr. Hamilton, I have a question that is actually was posted, and I had it myself when I was listening to you. I was surprised to see that the chemotherapy, you listed the chemotherapy as the third line for ER positive, HER2 negative population before one of the ADCs approved for ER
Correct.
positive, HER2 negative. What is the reason for that?
So it's because it's not approved first line.
Oh,
All the trials, you had to have chemo before you got an antibody drug conjugate. Now, those trials are happening right now, right? That's the TB01, TB02 that I was talking about, etc. They're gonna be moved up, but right now, the only place that they've been tested is for people that have already gotten chemo. And so for hormone receptor positive, we typically do endocrine as long as it works. Some people that's 2 lines. Some people it's 4, right? Then typically I would say capecitabine, Xeloda tends to be the oral chemo. I like to call it the gateway chemo drug. You can take it at home. It doesn't cause your hair to fall out. And then I'd be thinking about antibody drug conjugates.
Even you think after the trial results come out, you still think that you will?
Well, it'll be
capecitabine first.
It'll be a while before the approval changes.
Okay, thank you.
So we have a side effect management question that came up, and what is the best way that you manage fatigue?
It's a fantastic question. If I had the magic solution for fatigue, I'd be really rich and really famous. Varied. So the problem is fatigue comes from a lot of things, right? Fatigue comes from cancer itself, right? Like your body is like waging this internal battle, fighting cancer every day. It makes you tired, right? The treatment causes fatigue, right? The mental experience of having cancer causes fatigue. And so sometimes it's tough to tease out exactly what's causing fatigue. If people feel pretty well, you start a new drug and they're just like, oh my gosh, I mean, I could just take 4 naps a day. Well, it's pretty clear that the drug caused the fatigue. And so that's where we typically use a dose reduction. If people just have kind of, fatigue daily and it's not changing, etc., sometimes that's just the cancer. And honestly, what happens is a lot of times people, they don't slow down for cancer. They don't have the luxury. They've got to work. They've got kids at home, right? And so, I mean, you can imagine your body's fighting cancer and you're trying to be everything for everybody and you're just tired, right? There is a trick that is new, has a newer indication, Ritalin. So ADHD drug now has an approved indication for cancer-associated fatigue. And some people do really well on that. I typically do a pill in the morning, a pill at lunch, nothing at dinner. And some people get a little energy from that. So that's something that I've been using and actually find that older women do really well with it. I mean, you think like, oh my gosh, you're going to put a 70-year-old on Ritalin. They actually do really well. So that can be an option.
It's actually funny that you say, because anecdotally, Kate and I would agree with Ritalin for the longest time in our support groups. Older population seems to do very well on the Ritalin. So it's good to know that it's now officially approved. So I have a question, and I don't exactly know how to phrase it correctly, but basically we have we always get a lot of questions about lobular cancer because, as you know, lobular cancer is harder to see, it's harder to diagnose, and in the metastatic setting, it's a problem. So the question that somebody has asked, do they break out patients with lobular cancer in the trials for these for ADCs? And do we have any information to know which of the ADCs do better for lobular?
No, they don't. You know, we know that lobular has unique disease characteristics. It's more likely to be bilateral. It's sneakier and has fingers instead of growing in tumors, et cetera. There's kind of this idea that it's less chemosensitive, and sometimes that's true. But sometimes I see people kind of overstate that of like, well, it's not as chemosensitive, so I'm just not going to do chemo. Like, no, I mean, it's still recommended that people get standard treatments with lobular. On clinical trials, they always collect whether cancers are ductal or lobular, but there doesn't tend to be a whole lot of trials that are just for ductal or just for lobular. And so because lobular is less common, most trials are mostly ductal with some people with lobular. There's no reason to think that ADCs work better in ductal than lobular, though. There's a question in the chat about patritumab. Is it just in clinical trial or is it available now? It's currently just in clinical trial. It and Dato are currently just in trial, not available right now.
Are they still enrolling? Can people enroll?
Sure am. Yes.
Are you a principal investigator on both of them?
Yes.
Of course.
So we had a question submitted that talks about prior treatment with chemotherapy, and is there a thought on the effective response from an ADC?
No, I mean, you know, we have a lot of questions about sequencing ADCs. You know, I mean, we'll be the first to tell you we don't have all the answers, right? When drugs are new, great, they're working. But in terms of, well, what should come before them and what should come after them, it takes a while to figure out that sequencing. So we don't know if one ADC should be used before another because it works better in one order and doesn't work as well as the other. I don't think that chemo is going to make an ADC work better. I have to say, if I had any concerns, I would have a little bit of concern about pulling, you know, the big hitter out first. But on the other hand, sometimes people don't get to fourth and fifth line therapy. Something happens and they end up with liver problems or lung problems or something else. So I definitely feel strongly that antibody drug conjugates shouldn't be used before endocrine therapy, because in general, endocrine therapy is better tolerated and people can be on it for years and years before they ever need an antibody drug conjugate chemo. So you want to exhaust your endocrine therapies first. And we do know that once we treat with a bunch of chemo, when you go back and use endocrine therapy, it doesn't work very well. And so that's my theoretical worry, is as we bring ADCs up sooner, you know, well, we're probably going to see that those cancer cells that were able to survive those, you know, are uglier and probably don't respond as well. But in general, the rule of thumb is still use the best drug that you have available at the current time.
That makes sense. And it brings you bringing up the sequencing, and I wonder, so we had some information coming out at ASCO which talked about the dynamic HER2 status of TNBC and using that rebiopsy. So now we have some people that were possibly already on sacituzumab, and then, you know, now this data comes out and is saying, oh, you know what, let's rebiopsy and see if you do have that HER2 low. In that case, you know, can you follow up that ADC, the sacituzumab, can you follow it up with in HER2? I want to say in HER2 because I want everyone to understand, our patients actually, everyone here understands that brand name. So both Trodelvy and HER2 are approved, and they're approved in either order. And you most certainly can do one and then do the other, or vice versa. And they can go right after, sequentially, right after.
Yeah.
You would do that.
We don't have any data that that's the way we should do it, but we don't have any data that's not the way we should do it. And we do know that by themselves, they're both better than chemo. So as opposed to inserting some chemo that we know is less effective than the ADC, sure, I would sequence them.
So I wanted to ask you something that you and I had talked about a while ago, and we just did a webinar on the report back from Vasco, and the presenter talked about this too. The antibody drug conjugates consist of 3 parts, and the 2 parts are very important, right? The chemo and the monoclonal antibody. So at one point you told me that it's still not clear whether the monoclonal antibody part actually has any effect on the outcome, on the outcome of this particular drug. Do you still feel the same way?
So it's not that the antibody isn't important. It's that the antibody isn't what's giving you activity. So follow me here. It's a little complicated. So a tyrosine kinase inhibitor, right? Lapatinib, neratinib. The molecule is binding HER2 and it's stopping HER2 signaling in the cell. That's how it works. There's no chemo. Like, it's just stopping signaling, right? An ADC, on the other hand, it's not the HER2 antibody that's doing the work. It's the chemo. But the HER2 antibody is the honing mechanism. It's the carrier pigeon that's dropping the letter off at the right location. So you can't do it without that antibody, but you're not getting activity, I don't think, because you're stopping HER2 signaling or you're stopping TROP2 signaling. If you were, people with high TROP2 would do way better than people with low TROP2, and you don't see that. You're just using TROP2 to have the chemo delivered to a cancer cell, and then it's working like chemo, right?
Right, but on the other hand, we see that with the HER2 positive population, they do better, right, in trastuzumab deruxtecan.
But that's because they have more HER2 on their cell surface to attract the drug, right?
You think that's why it is?
Yeah.
Because, it's my understanding, again, I'm not arguing with you, that sometimes the molecules break off. And
Yeah.
I'm wondering if the antibody alone is actually doing work. It might be, right?
Yeah.
Thank you.
I think we have time for another question. So we have someone asking about TDM1, and it's working well for the body, but then it looks like developed some brain metastasis. At what point would you change drugs? Would you treat the brain metastasis,
Yeah.
stay
So
on the
that's
TDM1, or change?
Really tricky question. And we're like, I don't know. I don't even know if I do it the same for every patient I see. Essentially, it's tricky because if you have isolated brain spots and we treat them with radiation, and then we don't have anything active, sometimes we stay on the current therapy. We stay on TDM1. But if it really looks like the body's not a problem at all, and it's the brain that's causing us the most problems, that's when I think, maybe we should switch treatment to one of the 2 drugs that we know has really good brain activity. And that would be the tucatinib combo or Enhertu. And so, before we didn't switch therapies a lot because we just didn't have anything good. And now we have 2 pretty darn good drugs that actually improve overall survival for people that have brain disease. So if the brain's a problem and it's not one, little spot that was radiated and it looks great now and it's been stable, if it looks like the brain's progressing, then I would probably switch to one of the brain active drugs.
Okay, thank you.
Hey, can I ask one last question? I think
one last question.
It's a very quick question, and it came on in the chat, and I think it's worth asking. So somebody is asking, how does one get into clinical trial? Not everybody is treated in a National Cancer Institute trial, assignated cancer centers.
It's kind of thought that academic centers have trials and communities don't. That's actually not true. There's plenty of academic places that don't have very good trials. I'm actually in the community. I'm at a community practice and we have tons of trials. So probably the best idea is to ask your physician of, hey, where's my nearest clinical trial center? Can I get a second opinion there? Or Google online and look up breast clinical trials with your geography and schedule an appointment. You don't have to have your physician refer. I have plenty of people that walk in my door because they decided to come see me outside of their physician, and that's fine too.
That's very good advice. Thank you so much.
Thank you, Dr. Hamilton, for this informative program, and thanks to all of you for participating and submitting questions. Make sure to check out SHARE's website for upcoming educational programs and support groups. And don't forget to follow us on social media as well.
And I just want to say thank you so much. This, in my opinion, this was one of the best ADC presentations I've ever heard, and I've had a lot of people talk to me about ADCs. So I hope that everybody could follow it. And if you couldn't, we will turn it into a podcast, and I will try to break it down and make it sound a little less complicated, which I don't think it was. So please take a moment to fill out the survey at the end of the webinar. The survey will show up in the browser when the webinar ends, and the link will also be in the follow-up email. All surveys are anonymous. And that concludes our webinar, and we want to thank Dr. Hamilton again for a wonderful, wonderful webinar. Thank you.
Thank you.
Thank you so much.